Hello, I am Jessica Bridges-Weaver, Scientific Officer at Bioagilitics, where I focus on supporting clients in bioagilitics operations within our CMC team. I would like to discuss how we use flow cytometry as one of the platforms for potency determination for advanced therapy medicinal products, commonly referred to as ATMPs. As you are aware, flow cytometry is a powerful tool for quantifying cell phenotypes, viability, functionality, and protein expression. This tool is critical for assessing the potency and quality of ATMPs, such as gene therapies. There are situations in which the application of flow cytometry may offer discrete advantages over traditional and/or matrix approaches for potency assays. The platform's ability to generate multiple data sets simultaneously becomes powerful. For example, protein expression and cell viability in one assay. Within this poster, we'll share a couple case studies to demonstrate the application of flow cytometry to phase-appropriate development and validation of potency assays for an mRNA vaccine and AAV protein replacement therapy. In the first case study, the objective is to evaluate and compare the effectiveness of mRNA LNP delivery in driving a targeted protein expression using complementary analytical techniques. In this example, flow cytometry and mass spectrometry. Flow cytometry will assess protein expression at the single cell level, and mass spec was utilized to perform peptide level quantification for proof of principle and an orthogonal data supporting the flow cytometry assay. Figure 1 shows the workflow of the matrix of assays. In addition, Table 1 and Figure 2 depict representative data from both the flow and the mass spec. Data is presented as a function of the LNP mRNA dosing, and both methods show a discrete dose response in the signals. The flow cytometry provides a readout with strong biological relevance, while the mass spec provides a detection method for expressed protein that overcomes the need for targeted reagents such as custom antibodies. Flow cytometry itself overcomes the challenge of multiple or diverse protein expression. The combination of these platforms provides complementary insights to mRNA LNP delivery and protein expression. Flow offers high throughput functional assessment at the cell level, and MS confirms the quantities of protein expression at the molecular level. In the second case study, the objective was to develop a potency assay or matrix of assays to qualify the activity and expression of protein to demonstrate the MOA of a therapeutic. The flow cytometry was an ideal candidate because of the ability to fold both expression and functional attributes into a singular assay execution. Flow was used to determine expressed protein and functional outcome. In addition, flow is a logical complement to PCR used to monitor translational processes. The amount of expressed protein was determined by Western blot or in-cell ELISA. Transcription of the DNA was also measured using PCR, and these workflows are depicted in Figure 3. Figure 4 and Image 1 visually represent the expression and functional data. In Figure 5, the graph demonstrates the cell survival recovery is strongly correlated with viability. So in conclusion, flow cytometry is a cutting-edge platform with multicolor capabilities allowing for extreme flexibility. It allows for sensitive analysis of cell population, custom solutions for specific cell types, and therapeutic goals. Flow cytometry assists in overcoming the complexity of ATMPs by providing a tool to accurately analyze and measure the function of a drug of interest with high throughput in a short time frame. Bioagilitics provides tailored regulatory compliance solutions to ensure robust potency determination by flow cytometry as well as other platforms. Thank you for your time, and stop by booth 26 for more conversation on utilizing flow cytometry to support your potency program.